Right. Okay. How are we guys doing? How are you guys doing? I'm doing good. I've just been to B&Q to look for some stuff for the plants, my lovely little plants. Today we are going to be doing just a general overview of biology, but I want to make you guys aware of a very interesting day that is coming this Saturday before the first biology exam. What I have is a predicted paper. I bought it for like literally £1. It wasn't much, but I bought it. I have it sitting there in my emails, just waiting to be opened, and I thought we would do the predicted paper 2 days before the exam, so on the Saturday. Or maybe the Sunday. Maybe we'll do it on the Sunday. Because a predicted paper will be a really good way to test everything I've learned, because I am a very biased learner, unfortunately. I have aced a lot of exams this year thanks to really good luck. And what I mean by that is the questions that came up in the exams that we've done, I've revised before. Like, I've looked at the past papers that the teachers got them from. So I've had a very biased learning experience in biology so far. But that is all probably going to change. What I have here is some stuff that we did last stream, and my god, was it difficult. A lot of stuff on DNA, which I could not stand. This is the thing about exam questions, to be fair, is how difficult some of them are. I think it's almost a trend at this point that in every exam, you will always get one section which just makes no sense. One section which is just utterly baffling. And I like to kind of put reasoning forward, and I think I can do that here in the sense that if in the exam we do get a really obnoxious section on something which barely makes sense, just spam keywords. That's literally a guaranteed way to at least scrape a couple of marks. Just waffle and make sure to include as many key points as possible from the topic. So if it's asking about DNA, you can kind of branch off there. You can talk about, DNA, the enzymes related to it, talking about the complementary shape, the hydrogen bonds, the phosphodiester bonds. You can just ramble about that. That's one of the best ways to secure marks. If you don't know what you're talking about, just ramble and include a bunch of keywords. The reason I mentioned that is because today we have been given a bunch of past papers. Now, I actually told the teacher that I've already done these, but I'm not sure whether I have fully. So these are the June 2022 papers. And what I thought would be cool is if we unpick the staplings, like we always do. These are quite well lodged in. I think I have a stapler remover, actually. Don't ask why, I just have really random stationary items in my desk, but there we go. Today we're doing a past paper, paper 1. Now, here's a unique thing, right? The unique thing is we are at a point now where we're about 80% done with biology, right? We've done the biological molecules topic. I understand most of that very well, except DNA, shit topic. Then the mass transport we're still doing, but we've done digestion. We've done the gas exchange. We're just on mass transport now. We've done everything cell-related, immunology, things like that. The only thing we have not done properly, well, we're still doing, is we've done DNA, but we have not done evolution, I'm pretty sure. I think we have 1 or 2 more lessons on that to do. Okay. Honestly, I was gonna say, would it be worth doing paper one instead? I don't know. Let's just wing it. And this is already a lovely question that I'm seeing here. This is a lovely question. One of the infamous GMC Arts revision questions. One that we've done many a times. This is a bunch of diagrams. I hope you guys can see that alright. Right, what are these? Firstly, that is alpha glucose. The reason you can tell is because the H is on the top instead of the OH. I always draw them in order of beta first. So I'll always do OH, H, beta, and then obviously H, OH, alpha. That's just an easy little way I do it. So, that's alpha glucose. That is glycerol and 3 fatty acids. So that's going to obviously be a triglyceride. That is thymine and cytosine. So that's a nucleic acid. Alright, I guess that would be a strand of... those are 2 nucleotides. So I'll just put nucleotide. The reason I'm doing this is just to remember them, by the way. That is obviously beta-glucose. And that is just an amino acid. You can tell because it has the, what do you call it? The amine group. It has the amine group, it has the carboxyl, has the variable R group. Okay. So, what do we need to know these things for? Okay, let's have a look. Complete table 1 by writing. Okay, which one is hydrolyzed in the ileum and a product of this hydrolysis is found in my cells? So, my cells, remember, they are basically vesicles made from bile salts. So basically what happens is bile will emulsify the fats. It will break them into smaller droplets in order so that your lipase enzymes can act, breaking down the lipids into fatty acids and glycerol. You will then have the bile salts surrounding the fatty acids and the glycerol in order to bring it into the ileum to then be used. So that's obviously going to be a triglyceride, which I don't recall the number of. B. Number B, great, okay. Is formed by a condensation reaction between 2 alpha glucose molecules. I think that's a really weird way of just saying it's alpha glucose. 2 alpha glucose molecules, that's number 1, that's number 2. It has to be A. Is formed by the action of DNA polymerase. That's a lovely one. Obviously, it's C, the nucleotide. Gives a positive result in an emulsion test. Remember, emulsion test, you add your, what do you call it? Ethanol? Is it ethanol? Probably ethanol. You add your ethanol, you shake it. You add your water, distilled water, shake it. And a cloudy white emulsion will form if there are lipids there. And remember, an emulsion is just a droplet of a liquid suspended in another liquid, I'm pretty sure. So that would, again, be B. Now, that is kind of... I love when they do this. They make it so that you can really easily get tricked, right? Look, A, B, C, D, or E, but we've only used 3 of them there. There's still D and E. People might fall for that. People might be like, hold on a minute. If that's B, that can't be B. It's a common trick. You have to read it carefully. It may be used once or more than once. Typically, if it says may be used once or more than once, you'll likely use it more than once, otherwise I wouldn't have put it. Alright. Describe the mechanism for the absorption of amino acids in the ileum. This is your co-transport. And I'm not too confident on this topic because I feel like it drags. So let's let's just go here. So this is going to be your small intestine, your, I guess, just amino acids in here. Then you have the cells lining the ileum. And then you have obviously your capillaries. So, this is kind of a multi-stage thing. Firstly, you will have sodium will move via active transport through a sodium-potassium pump. This causes a low concentration gradient to be created, which forces the sodium ions to move in via facilitated diffusion. This also causes the amino acids to be brought through with the sodium ions. From there, the amino acids will travel through simple diffusion into the capillaries, and the sodium will, of course, join back up. That's like a very simplified explanation there. This is 4 marks, so I want to get a couple here. So firstly, I'm going to say Na plus ions travel via active transport. Okay, I should probably specify Na ions in the ileum. So Na plus ions in the ileum travel via active transport through the NAK pump. This causes this creates a concentration I don't know what to specifically put here. This creates a concentration gradient between the small intestine and the ileum. Okay, so because of that, okay, I don't know the full scientific terminology here, so all I put was sodium ions in the small intestine move via facilitated diffusion into the ileum and bring amino acids with them. I'm not sure whether you would need to talk about a channel protein there or something. I talked about facilitated diffusion. I'm going to assume that's enough, but I'll add a slight question mark there. Amino... Alright, so here's the full extent of what I've wrote. Sodium ions in the ileum travel via active transport through the NAK pump. That's what I've put there. This creates a concentration gradient between the small intestine and the ileum, So sodium ions in the small intestine move via facilitated diffusion into the ileum and bring amino acids with them. The amino acids in the ileum will then travel via simple diffusion out of the cell and into the capillaries. I am unsure whether I have put too much detail or too little. I mean, it's 4 marks, and I've put 4 points there. So I'm going to be real and say, I think maybe that could be 3 marks there. I feel like it wouldn't be fair, purely because I'm not too confident with this topic. I think there's something I've missed. I feel somewhat confident with that though. I think I'm going to pop a prediction there. I'm going to put 3 there, and I think that could easily be a fair mark. Okay, give 2 types of molecule from which a ribosome is made. This is such a lovely question because everyone gets it wrong. I'm pretty sure it's made of RNA, and I think you can get away with saying proteins. I don't know if you need to put mRNA or what, I just put RNA. Complete the table to give 4 structural differences between a DNA molecule and an mRNA molecule. Okay, 4 differences. Alright, can we draw this? Let's draw this. Piss off, alpha glucose. Okay, let's draw this. Deoxyribose nucleic acid. I forgot what these look like. Your phosphate group, your base. Okay, so that's deoxyribonucleic acid, and then your mRNA is basically the same, except with a couple key differences. mRNA. Bear that in mind. All right. Right, let's get working. So, DNA, your first difference is that is deoxyribose. That is ribose. In terms of bases, is it adenine? AUCG, no. ATCG. That could be thymine. That will always be uracil. If it's thymine. What are the differences? mRNA. So surely it would have its introns removed. Really simple differences. Hold on. The scale. These are smaller. And they are single-stranded. Meanwhile, these are fully double-stranded. I think that's our differences. So, DNA made of deoxyribose, made of... See, my other difference I was going to put was contains introns and extrons, only contains extrons. Okay, non-coding DNA is removed. So DNA can contain thymine replaced with uracil. Larger smaller than DNA, single-stranded, double-stranded. Cool. Okay. So those are the differences. I am writing using Sharpie, so I have kind of fucked up. But okay, I think I got that question right. And I think this question, I also probably got right. I'm going to be optimistic here. That's 1 page done. Cool. Let's keep it moving. Let's keep it popping. Popping shots at the back of a shop. Walmart blocks. I don't know. That's a song I wrote. Featuring Unity. Oh, we've done this so many times. We've done this so many times. I kind of can't be bothered to do this again. This is a question we've done like 20 times. This was in our mock exams. We've done this in like an after school revision session, a holiday one. I've done this many times on stream, including the night before the mock exam. I can't stand this question. It's just boring as shit. That is a flagella. TEM microscope versus SEM. Okay, very quickly. The differences. Transmission. Scanning. Okay. Very quickly. Oh, the stream is having a technical issues. Shit. Is that good? We good, YouTube? We good? YouTube? One sec. Having some technical issues here. Well, I reduced the stream quality. Hopefully you guys should see a little... it might be a little less clear now, but you guys should not have any lag. Okay, look. The gimmick here, transmission, watch. You have your little specimen. Watch. Transmission goes right through it. Scanning hits the specimen and then it bounces off and is picked up by an electron detector. Do you get it? If you do, congrats, because that's literally all this boils down to. And you can use interpretation to really get the rest. So in scanning, that's going to produce a 3D image. In transmission, a 2D. Because it's going right through the specimen, it's going to have a higher resolution compared to this with a lower resolution. Transmission has a risk of damaging the specimen and causing visual artifacts. No other ones. Indiana Jones picks up. Not really true. It's the whole idea of visual artifacts. It's like, I don't know about tonight, dude. My MacBook, I'll have to get it all set up and stuff. Might take some time. We could do it like next week or something. I don't know. I mean, there's no rush. There's no rush or anything. We have literally until mid-August to get it done. Visual artifacts. The best way I can describe artifacts is, imagine you're like making pixel art on Minecraft and you use like a Google image. You paste it into the world, but you can see like the edges and it looks really blurry. And it's little like patches. That's basically what visual artifacts boil down to. So I did all of that for a 2-mark question. I'm gonna kill myself. Figure 2 is different from an image of a bacteria obtained using a TEM. So it's basically saying what's different between a scanning. Description. A 3D image. So SEM produces a 3D image as electrons bounce off specimen and are used or are detected by a computer to construct a 3D image. Okay. I think that's about right. Honestly, I always get this question wrong. I kind of struggle to explain it, even though I know what to do. Scanning electron microscope produces a 3D image. Maybe I should say TEM. Oh, hold up. It says a difference. I feel like I've got that one wrong because I didn't really describe a difference. But you know what? We'll stick with it because, to be honest, I've done this question so many times, I'm sick of it. The resolution of an image obtained using an electron microscope is higher than the resolution obtained using an optical microscope. Remember, optical microscopes use light. The wavelength of light is very big. Meanwhile, the wavelength of electrons is very small, allowing for a higher resolution. So, light microscopes have a lower resolution as wavelength of light is much greater than an electron. That's basically what it boils down to. That's basically what the gimmick of that is. I hope chat is working and you guys can see the stream. I'm gonna go open my blinds a little. Quick intermission. I want to show you guys my chili pepper plants. Look at how cute this is, right? Look, my plant. It's so cool. It is my plant. When I first got it, it barely grew past this sign, right? Barely grew past this sign. And now look, it's massive. It's a massive plant. I will treat this thing like my child. I'm looking forward to it, to be honest. I mean, yeah, this thing is going to be sick once it's fully grown. I was watching a video where it explained, but it was like a time lapse of chili peppers. And it showed that once it reached about this size, maybe a little bigger, that's when it started to flower and eventually have the chili peppers. So what I'm really waiting for now is just to see where the flower kind of spawns in at. I mean, there's kind of a growth there at the very top, like a couple little leaves, and then you have a couple leaves there. Very cool. I love this plant so much. I'm going to grow the greatest chili peppers ever. And yes, I do water them. They are very well watered. Probably better than me. A student determined the size of a cell structure from a photograph obtained using a microscope. He used a ruler and a calculator. So, measure the actual size. Sorry, measure the image size. Bacteria in millimeters. Multiply by 1,000. It's IAM. We're working out the actual size. Yeah. So, and divide by the magnification. Okay. Name 2 structures found in all bacteria that are not found in plant cells. I love this question because I get it wrong. First one is 70S ribosomes. They have smaller ribosomes. They have to have ribosomes in order to synthesize proteins. That's number 1. Number 2, I'm not sure. It's not a flagella. It's not plasmids. It's not those little hairs. Not found in plant cells. Circular DNA? That's my guess. Name 2 features of HIV particles that are not found in bacteria. Lovely question. You got your protein code or your capsid. You could put lipid envelope as well, maybe. I'm gonna put the goat of biology, reverse transcriptase, enzymes. There you go. Literally the goat of biology. Ooh. Oh, okay. We're on to territory I've never done before. Okay, so lugworms create tubes in the sand on seashores. The tubes are filled with seawater. Okay, so we have to assume this is all full of water. A scientist measured the partial pressure of dissolved oxygen in seawater at different places. The partial pressure of dissolved oxygen in lugworm blood is that. Using data from figure 3, what can you conclude about the uptake of oxygen over the entire body? Okay, this is kind of a tricky one, I'll be honest. I don't really understand. So, you're getting about 20 kilopascals of water coming in. You're then getting less water coming out. So, the surface with the gills. So it seems to me as if the lugworm... I mean, right there, it's 17.6 kilopascals, right there. By the point it reaches the end of the gills, it's been reduced by about 10 kilopascals. So what can we say? When the water reaches... So when the water reaches a lugworm, some of it, the oxygen, diffuses into it. As it reaches the end of the gills, should I put around half? What can you conclude about the uptake of oxygen over the entire body? As it reaches the end of the lugworm, is absorbed, meaning gills absorbed most of the O2. I don't know what this is asking. I'm genuinely confused here. What's it asking? What can you conclude? I don't know. Like, this is very big. I guess what it's trying to imply is the lugworm absorbs a little bit of a water here, like about 2.1 kilopascals there. It absorbs about 50% there. So that would be 8.8. It absorbs about 8.8. And then there, it absorbs 0.5. So the vast majority of oxygen intake is occurring... Okay, here. Gills absorb... Most of the O2 uptake occurs in the gills, but the ends of the worm also intake O2. That's kind of what I would conclude, but that's that's a difficult question. Because I know what it's asking, I get what it means, just literally stating how the pressure changes, but how would you put that into words? I don't know. It's weird. Right. I think we'll stop after question 4. So we've done 4 questions out of about 8. That's very valid. That's very, very valid. Okay. How are we going to do this? We'll stick this one there. Oh, 3 marks, and it's a calculator question. The oxygen saturation in the blood of a lugworm is 92%. The lugworm has 0.2 centimeters cubed of blood. Calculate the volume of dissolved oxygen in the blood of the lugworm using this equation. CDO is a concentration of dissolved oxygen in the blood, with units centimeter cubed oxygen per centimeter cubed of blood. What in ever the fuck is this asking? What in ever the... Okay, hold on. 2.7. What is this asking? What the fuck? The oxygen saturation in the blood of a lugworm is 92%. The lugworm has 0.2. Calculate the volume of dissolved oxygen in the blood. The concentration of dissolved oxygen in the blood. Would you literally just do 92% of 0.2? Is that seriously what it's asking? I mean, you put it in a formula? No way that's what it's asking. I'm sorry. That cannot be what it's asking. Because that is obnoxious for how many marks this is. This was worth more marks. This was worth nigh as many marks as that dumbass equation question. Great. I'm actually going to cry. Is this genuinely the question? 92% times 0.2. 0.184. So 0.184 divided by this obnoxious number. Great. If this is actually the answer, it's over. It's over. 0.184 divided by 0.000031. 59. Jesus Christ, that's a huge amount. The intensity of the red colour in blood is affected by the partial oxygen pressure of the blood. The intensity of a colour in a solution is measured using a colorimeter. Okay. The scientists use a colorimeter to measure the intensity of the red colour in samples with different partial oxygen pressure values. Okay. She preferred a calibration curve. Describe how the scientist will use information from the colorimeter and her calibration curve to determine the PO2. So, sample use a colorimeter on the sample of lugworm blood. On the curve. I don't know how you would explain this. You would like just follow the line to see what. And follow the line. I'm gonna be honest, I had no idea what I was saying there. Right, shall we just get this fucking marked and done with? Because I can't be bothered for this shit. Some of those questions made utter no sense. Okay. Pretty sure this is missing, like, multiple questions. Okay. Right. Let's get this marked. Well, the things that we've done, let's get them marked. Okay. Here we go. B is hydrolyzed in the ileum. Nice start. A, C, B, lovely. That is a lovely 4 marks. Okay. Na plus ions travel via active transport through the NAK pump. Co-transport. Sodium ions actively transport from cell to blood/capillary. I should have specified there into capillary. Creating a sodium ion concentration gradient. I did put concentration gradient. Creating a sodium ion concentration gradient. Facilitated diffusion of amino acid. Okay. So I should have really said, so sodium, okay. So this is what it's saying. Facilitated diffusion of amino acid into cell when higher concentration in lumen. So, fuck. I don't get this mark scheme. I don't understand this. I don't really get this. Max of 1 mark if ions not mentioned. Accept Na+, accept pump for transport. Accept diffusion via channel. Okay. So I feel like I might have gotten 2 marks there. Maybe 3. If I had put co-transport at any point, that's just 1 mark. Holy shit. That is fucking disgusting, to be honest. You could get a mark just for putting co-transport with no explanation. Facilitate diffusion of the amino acids into cell when higher concentration in lumen. Co-transport. Sodium ions actively transport from cell to the capillary. So I think I just went out of my way. I gave a very long-winded explanation of the sodium ions involvement. I should have really added a tad more about the amino acids themselves. I should have talked about how facilitated diffusion of amino acids. Honestly though, I could have easily gotten 4 marks on that. Had I literally just specified a tad more, I could have easily gotten 4 marks. And that was the most obvious mark ever that I was gonna put, but I thought it was too obvious. RNA, protein, easy 2 marks. Okay. DNA has deoxyribose, mRNA has ribose. I guess I should have just put has, not made, but okay. DNA has thymine, mRNA has uracil. So I guess instead of giving specifics, I could have literally said made of. Or has. Sorry. Okay. DNA is long, mRNA is short, so I put larger and smaller. I'm sure that'll count, but I'll just put long and short in notes. mRNA has no hydrogen bonds. Double-stranded, single-stranded, lovely. No mention for introns. Great. I'm glad I didn't put that because I could have. I nearly talked about introns there. So I'm gonna give myself 4 marks with that. So that's 6. So that's 12 marks currently. Flagella, 1 mark. 3D with SEM, but 2D for TEM. Well, that's what I put. Because electrons deflected / bounce off. I put electrons bounce off specimen and I detect. So I gave a very long-winded answer, but I'm sure that's right. Okay. Light microscopes have a lower resolution as wavelength of light. Okay, light microscopes have a lower resolution as wavelength of light is much greater than an electron. Longer wavelength. I put much greater. As wavelength of light is much greater. I'm gonna give myself that. I think that's right. A student determined. Okay, measure the length of a structure. So measure the image size of a bacteria in millimeters. Multiply by 1,000 and divide by magnification. I'm gonna say that that's right. 70S ribosomes. Oh, come on, the most obvious one. I did get it right, but I could have put murine cell wall. Easy, lovely. Capsid reverse transcriptase. Okay, hold up. That is 3 for 6, 10. 10 marks. Okay, I basically cleared that section, but let's not get ahead of ourselves. This is a nasty old section. I don't get this. Enters by diffusion. Down. Oh, come on! 1 mark for fucking mentioning the concentration gradient. Why didn't I put that? Okay. This is the mandatory bit where I refer to what I said at the start and ignore it. If you don't know what you're talking about, spam keywords. I could have easily put diffusers through a low concentration. I did talk about diffusion. Most across part of body with gills. I put most of the uptake occurs in the gills. That's a mark. Gills provide a larger surface area. I wouldn't have fucking known that. 8.8 kilopascals over gills. So you could have included data there. 8.8 there and 2.6, which I did number. I did write that on the diagram. So I'm going to give myself 2 marks, which is better than I thought I did. I thought I wouldn't have gotten any. So in that question, what I would have needed to specify is I should have immediately identified, but it's a diffusion question, okay? Here's what you do if you don't know what a question's asking. Try and identify what it's asking. For example there, it's talking about dissolved oxygen, the uptake of oxygen. It's clearly a diffusion question, or at least a transport question. If it's talking about transport, always mention surface area to volume ratio, concentration gradients. Okay. I'm honestly a little shocked I didn't get that. Like, as in I didn't realise that that's what I had to put. But hey, little mistakes that can be corrected. We're literally just talking. Oh, how did I get that? I got a positive number. I got 6,000. This got 0.00093. You had to read the graph, rearrange the equation. But you had to rearrange. What? Okay, this is genuinely what I mean when I say a vast majority of marks that I lose in these exam questions aren't due to simple mistakes. Some of them are. A vast majority of the reason why I lose a lot of marks in these exam questions is because of overly complicated math that I don't understand. Like, how would I have known to rearrange the formula? How would I have known how to do that? You would have had to, like, interpolate the graph. To be honest, I did notice, to be fair, I did think maybe this is too easy for 3 marks. But I never once really thought that that's what I would have had to do. Although, I'm looking back, that answer is really weird. So no, fuck off answer. We're not doing that. Final one. Measure the light absorption, interpolate, draw. Bro! I was gonna... Oh, no! I was gonna put interpolate. I was literally going to put interpolate, but I thought that wouldn't have counted because biology is weird. Bro, I'm gonna kill myself. This is awful. Use colorimeter on sample of a blood. So what it was asking for there was measure the absorbance. So basically what I put, but more specific. So no marks for those, so on. So 5. I'm just gonna count how many marks I could have gotten. Well, 5 plus 10 plus 4. I've not done very well here, have I? So out of 33, and I need to see how many I've got now. 33. Out of 33, I'm going to probably bet 22, so about 66%. I got 12 plus 10 plus 2. 24 out of 33 or 72%. That's almost 3/4. That's almost 3/4. That's almost 3/4. I'm happy with that. I'm happy. Yeah, looking back, I could have easily gotten 1 or 2 more marks for pretty obvious things, now I think about it. That co-transport question, if I had literally put co-transport, 1 mark. Easy. I don't know how I really could have gotten the other mark though, so I'll be honest, that's probably the best I could have got, 3 marks. But, that question about the lugworm, it was very challenging, probably one of the most difficult ones I've done so far. We got 50% on it though, which is great. Thinking back, I should have immediately... Okay, let me give you guys a couple tips here. If you look on an exam and you look at a question and you're like, what the fuck does this mean? Try and identify at least the topic it's asking about. So for example, in fact, we could try this with another question quickly, if I can find one. Okay, hold on. For example, this question here about hemochromatosis. What the fuck does that mean? Well, if you look at it, it's asking about ions, blood plasma. So this is clearly going to be a question about, hemoglobin, likely. It's probably going to be talking about the role of ions in hemoglobin. So, you can easily identify things there. A table here about dogs with cancer? Such a weird question. But like, you know what I mean? If it's a question where you don't know fully what it's coming from, just state basic facts. Like that, diffusion. So obviously it diffuses. Okay, I'm going to ingrain this in my head now. When talking about diffusion, always to the concentration gradient. Right, that's your tip for the day, guys. If in an exam you get a question and you mention, even just in passing, diffusion, always refer to moving down the concentration gradient. You could possibly get a mark. And if it talks about describing a process, but it doesn't name the process, state the name of a process. Oh my god, okay. So that's the two things I learned today. Number one, always refer to diffusion and the concentration gradient when talking about the movement of molecules. And if you're given the name of an unknown process, state the name. Stay happy, healthy, and safe. That was somewhat tough. That co-transport question really got me thinking. Yeah, that co-transport question really did get me thinking there. I was kind of puzzled. And then that look one question we did relatively well on, considering I've never done that before. I've never done a question talking about, blood plasma and... Sorry, not blood plasma. I've never done a question where it's kind of like, you have to decipher the meaning. I was kind of unique. But, hey, we did good. We did alright. 75%. And that's after not many days of revision. I'd happily take 75 in the exam. I'd take 70, hell. I'd take whatever. I'll see you guys later. Hopefully you remember these exam things, because I will.